All right. I know this is the last talk on the last day, so thanks for sticking out and seeing what we have to talk about. So Virica is focused on overcoming the hidden barriers of cell defenses to viral infection. Really, when we're talking about cellular defenses, we're talking about thousands of genes that are in manufacturing cells, in primary cells, that basically prevent viral infection or viral production. As you probably know, there are multiple layers involved in that response, and at the end of the day, they lead to the expression of effector proteins that destroy viruses as we're making them in the context of manufacturing of viral vectors, or that will prevent efficient transduction efficiency if we're talking about cell therapy production. At the end of the day, all these pathways can really significantly impact your viral vector yield, quality, and impact your cost of goods, and at the end of the day, your likelihood of success at commercial stage. So at Virica, we've addressed this challenge by using a small molecule platform, and we've been developing the viral sensitizer technology for several decades now. And really what we have is a large collection of small molecules that are proprietary that essentially can be used to enhance either viral vector production or cell therapy production. The VSCs essentially enhance the infection or growth of viruses by transiently dampening these cellular defenses that I just talked about. And so, again, we have hundreds of molecules identified initially by high throughput screening. We can categorize them in two parts. Our first generation, which is really all virus types, and so this includes anything from influenza to AAV gene therapy. And then our second generation is really AAV and lentivirus focused. And again, these types of molecules can be used both to enhance virus production as well as cell therapy production. You might ask, why so many molecules? Well, the reality is in the cell and gene therapy space and even in the vaccine production space, there's an incredible diversity of systems out there. So it's really critical to have an array of tools to dampen the right pathways at the right time in the right situation. And so this is where a high throughput virology platform comes into play. So we've been developing approaches to look for small molecules that boost viral production for decades, as I said. And so we've more recently married this high throughput virology approach with a powerful statistical approach, which you probably are familiar with, which is design of experiment. And so what we've been able to do is actually take the power of the DOE framework, which is to really reduce the formulation space to something manageable, and then quickly screen through all the possibilities using high throughput screening. And, just to size the problem, if you're out there, you're a new therapeutics developer, either for AAV or Lenti or whatever it might be, just for the combinations of media, transfection reagents, and cell lines that are out there, there are over 19,000 different permutations of those things. So, what are you going to pick? You're going to go with your best guess, or you're going to try to test them all. Testing them all at scale would take you decades. And so what we're proposing here is actually leveraging this type of platform in order to really quickly screen as many possibilities as you can. Of course, we have also our VSC molecules that we can integrate into that workflow, and I'll show you some examples of that. Currently, our throughput is about 1,000 conditions a week. Of relevance to this crowd, obviously AAV gene therapy is a big market segment for us. Here's an example using some of our first generation molecules. This is a program that initially started with an AAV2 optimized process, then moved to AAV8 for the therapeutic product. They did some initial evaluations in the Amber 15 and then scaled to 5-liter bioreactor, and the yield improvement that they obtained with our VSCs was on the order of almost 4x, and importantly, an observation of improved full empty particle ratios. This program is now moving to 50-liter scale, or has validated at 50-liter scale with GMP grade VSE, and of course, residuals tested showed no residuals detected at the end of the process. For this particular therapeutic program, I think the cost impact is pretty significant. So in their baseline process, it would take them 3 batches to manufacture for their 36 patients that they wanted to enroll. We basically modeled for them using Biosolve the impact of yield improvement in the upstream in their process. And you can very clearly see that compared to the additional cost of the VSE, the savings are quite substantial. And so for producers that are entering clinical phases, addressing the problem at this stage and improving the yield, even by a factor of 2 to 4, can really have substantial, meaningful impacts. So I just showed you an example with the first generation molecule. We have been uncovering a wide range of different solutions and different molecules for AAV and lentivirus specifically. We recently established a collaboration with Rythera, which is a CDMO actually located close by in Rome. If ever you're interested in visiting them, let me know. I'm happy to connect you. They basically had 2 different compounds, a first generation and a second generation, both enhancing yield over 2x, in combination now reaching about 4x. All this translated in scale up at 2-liter scale. And really, they are now moving forward with this as an offering. Importantly, it's allowed them to basically enhance the productivity of their RICEL platform to be actually even better than commercial cell lines that are currently available, kind of avoiding those licensing fees that are associated to that. Okay, so that's the compounds themselves and what they can do. What about high throughput virology DOE at work? Again, this is where we're now combining as many possible combinations as we can, leveraging that high throughput screening. So this is an example that we did with another customer platform where we tested 6 different VSEs at 4 different concentrations each, basically doing all the permutations of that in high throughput. Again, we identified a formulation that led to substantial enhancement in yield, which we then validated at scale. So it's important to understand that our assays are actually transduction based. And I know that out there, lots of folks quantify AAV by VG per mil. But frankly, if you're treating a patient, you're not treating them with genomes, you're treating them with transduction units. And so it's a very important parameter to be looking at very early on. So what we find is that not only do we improve yield at the PCR levels and the number of genomes, but we can actually reach almost 5x improvements in transduction unit productivity. And so I think you can appreciate that's really substantial, and again suggests improved full empty particle ratios. Other things that we tend to observe with these types of molecules are actual reductions in host cell DNA contamination as well, again, owing to the productivity enhancement per cell. So one quick example for lentivirus, again, doing very similar high throughput DOE, we can obtain formulations that enhance yield pretty substantially. In this case, on the left, what I'm showing you is a plasmid-based transfection process in adherent cells. One thing that we noticed, and this was through a collaboration with Spherix, we actually looked at the amount of full particles in this using their platform, and what we observed is that not only did we have improved yield, but the amount of full particles also was substantially improved. On the right side, what you're looking at is a stable producer cell line, and so here again we evaluated a number of different compounds in a production system that at baseline was actually pretty productive, and we saw almost a 5x improvement in yield across a range of viral sensitizers. Okay, so that's the virus production aspect. I'm just gonna tell you a little bit about the cell therapy aspect of this as well. So as I mentioned, when you're trying to transduce primary cells with foreign genetic material, and particularly a lentivirus or anything like that, you're going to induce antiviral responses. These are typically immune cells that you're dealing with. They have very robust antiviral defense pathways that you should be dealing with to get the maximum transduction efficiency. So this is an example where we've used our VSCs to increase the transduction efficiency in T cells. On the left, you're looking at the transduction efficiency, so you're getting about a twofold enhancement in transduction efficiency and very minimal impact on cell viability. Yeah, just for the sake of time, I'm not going to show you all the data, but we're increasing our data set here, again, through a lot of collaborations that we've established, seeing transduction efficiency enhancements on the order of 2 to 4-fold, depending on the system, which we've observed so far across T cells, NK cells, mesenchymal stem cells, and hematopoietic stem cells. Okay, so just to finish off, again, we're working with small molecules, but again, just a reminder that this can have a big impact, particularly on your cost of goods when you have productivity increases, on the order of up to 7x. Very easily integrated in existing processes, and in many instances can actually improve the quality of your viral vector, as I showed you previously. Again, I showed you gene therapy examples, but we work with any type of virus, again, in any type of substrate, all the way from eggs for influenza production, all the way to AAV gene therapy, as I just showed you. So with that, I'd like to thank you for sticking it out for the last talk, and enjoy the reception, and please visit our website.